Please welcome Stanley Lapidus.
Thank you. Thank you, ladies and gentlemen. So I've been doing tech for 40 years and tough tech for 30 of those. And my own definition of tough tech is unlikely to work, takes a lot of time, and takes a lot of money. It's, I think if I were really smart, I would not have done this, but alas, this is what I do. One reason this is what I do is because it's enormously gratifying, and I'll talk a little bit more about that later. I've started 6 companies over the years. 2 were failures, 2 were modest successes, and 2 were quite successful. The modest successes were not tough tech, and they were modest successes and modestly gratifying. They, in the end, meant nothing to me, and the time I spent there was a training ground for what came next. In those years, what I learned was focus first on an interesting opportunity. I mostly work in healthcare. I do work exclusively in healthcare. Another caption for this slide would have been, find problems that are clinically important and scientifically overlooked. And this has kind of become my mantra. This is my operating plan. I look for markets that are huge, because huge markets forgive mistakes and forgive the fact that it takes forever and a huge amount of money. Clear value propositions, because I tried a business without a clear value proposition. Rodney Brooks earlier talked about a company called Cognex, which I think is the leader in industrial machine vision today. I started with my classmate of mine, a competing company called Itran, and I don't think I ever enjoyed a single day there. Things worked out okay, we made a few dollars, our investors all made money, I got to do it again. But it was the problem of explaining to the automotive industry why quality matters, this is in the 1980s, in an industry which never measured quality. In the US, GM was our primary customer, they only measured output. Your bonus never got dinged because cars had warranty repairs. If you were an executive, your bonus was solely dependent on productivity. So I learned quite viscerally that value propositions really matter. In healthcare, value propositions are measured by, did you save a life? Did you reduce pain and general misery? And did you save money for the system? Almost always in healthcare, value propositions are clear. And finally, because these things take so long and cost so much money, having strong defensible IP is really important. In industrial machine vision, in the company Itran, we were very proud to have our first patent. We had 200 competitors, and our patent blocked no one. And so it's not just having patents, it's having good patents, patents that really have blocking power. So the quick version of the story is we were on our way to sell Itran. 2 of my investors approached me. One of them is the firm now called Flagship Pioneering. It was then called Morgan Holland Ventures, and said, Stan, here's $100,000. Put in a little bit money of your own. We'll give you half the company if you want to do this. And if we like what you come up with, we'll put some more money in. So I think I spent the first 3 months being angry at my partners in the earlier venture, because it did okay, but not great. They no doubt felt the same way about me. But I came up at that point with this credo, these set of things I would look for in anything I did next. And pretty quickly, because I have this machine vision background, I was looking for problems in healthcare, because I like that, actually, in which machine vision could operate at scale. And one of the great problems was the pap smear, early detection of cervical cancer. It's quite a story in itself, but a guy named George Papanicolaou in 1928 first reported that cancer can be identified early at a cellular level by aspirating the vagina, collecting the liquid that comes back, putting it on a microscope slide, and examining those cells. The big dark nuclei, which everyone knew were a part of cancer, could actually be found on cells years before a woman had cancer. That lesion could be scraped out with minimally invasive techniques even then, and a life would be saved. And since cervical cancer was the #1 cancer killer in women, this was a big deal. Unlike any other lab test, which even from the 1950s on was starting to be automated, pap smears were entirely manually examined. So a cervical sample was scraped, smeared onto a slide, and then the slide was examined under a microscope, and still is, looking for big dark nuclei. By the 1980s, so 60 years after Papanicolaou's work, instead of the pap smear being hailed as a life-saving procedure, which it was blasphemed and damned because it was so prone to errors, more than any other lab test. If you look at a slide like this, and this is 1/100,000th of the field of view of a pap smear, a single slide, if you look at this, finding the big dark nuclei when they're obscured by blood and mucus is really quite difficult. But I thought of it as simply as a problem of inspection, a factory automation term. What we need is a better inspector. So the idea was, maybe I could work with my colleagues and develop an imaging system that would read slides and not make mistakes using a primitive form of artificial intelligence. I think the hardest decision I ever made was to say this technology with 1980s level Macintoshes wasn't going to make a difference. We could find cancer cells. We could find them, a lot of them. We almost never missed, but we were never as good as humans. And we were asymptoting to something below human performance. And the tough decision was the pivot here, the pivot from saying, let's just keep at it, throw more money at it, faster processors will crack the problem, into a pivot that said maybe the pap smear itself is the problem. We came up with a gizmo, I'll show you in the next slide, I think, called the ThinPrep, which reformatted the pap smear. So instead of having a gamish, a mush, a smear of cells, cells are laid down in a thin uniform layer, making their examination much easier. This is from the same patient. And I think none of us needs cytology training to say there's a big difference between the cells in the upper cluster in the top middle versus cells along the lower portion of the image. And this is the transformation we did. The amount of resistance that we received from the clinical community was really quite remarkable because it required retraining, it required learning how to reread a pap smear. And this was the tough tech part. Compared to the amount of time and money to invent, to build prototypes, the cost of clinical testing and getting the clinical community to adopt was probably, 100 to 1,000 times greater. It was millions versus hundreds of millions, high hundreds of millions before what finally happened. So conventional wisdom is always hard to overcome. In medicine, it's especially hard to overcome. George Papanicolaou was a revolutionary. The idea of early detection of cancer didn't exist before him. The people who trained with him and came after him simply had the view that the conventional pap smear was perfection, and any mistakes that are made are due to negligence or insufficient training. They never considered that the pap smear itself was highly suboptimal. The people who became our great allies weren't those people who, by the way, are the age I am now. They were the age then I am now. But people who were young physicians in their 30s, 40s, and into their 50s. They became our advocates. They spoke for us at our FDA panel hearing. And things turned good when finally we got a label from the FDA after many years of superiority to the conventional pap smear. So now if you didn't do a ThinPrep, the plaintiff's lawyers would be out suing you, saying you should have known this was a better technology and you didn't use it, and this woman got cancer because of your negligence. So in a perverse way, some of the bottom feeders, there's no nice way to say it, of our society became our greatest allies. The technology itself was simple. It's based on the French press, and I know this principle well because this is how we make coffee at home. And one morning, years before Cytic, I overfilled the glass thing and pressed very hard on the handle to try to get the coffee to the bottom, and the little thin glass thing, whatever it's called, canister, exploded, leaving quite a mess. And so the principle as I was cleaning up the mess was the pressure until the filter was clogged is proportionate to the coverage of the filter, and afterwards it's pretty much infinite. We adopted adapted it to collecting cells. If you think of a coffee grain as a cell, or a cell as a coffee grain, creating a thin uniform layer on a filter, and once you've achieved a certain coverage, you can stop, is the principle of the ThinPrep. People have tried to make these kind of preparations before, but the question of how many cells do you have was elusive, and this simple principle and a $10 pressure sensor led to a pretty good outcome. The instrument we developed looked like this. The instrument sold for $30,000. The filter and the reagents were sold for, I think, initially $9.75, cost about $1 to make. So we demonstrated that we could have improved clinical outcomes with gross margins and operating margins like the pharmaceutical industry. The company, let's see what we have here. Big reduction in screening errors, big reduction in unsatisfactory pap smears, and a very strong FDA label. The FDA started out not as our friend, but ended up playing a crucial role in the company's success. There's over 1,000 publications now. And the end part of the story is the company was acquired for $6 billion, I think it was 19 years after its founding, and 9 years or 8 years after the IPO. So it turned out well. It took far more time and far more money than I ever managed. I thought of this as 3 years and $9 million, and I wasn't even close. But because the market was large and because the value proposition was clear, investors cut the company the slack that it needed to disrupt cytology and to create a new standard of care. The thinking about Cytic was, and as the retrospectoscope began to make the picture clear, was that we weren't a laboratory equipment company, which is what we thought we were at the beginning. We were a women's healthcare company, or we were a cancer prevention company. Thinking along those lines, I began to ask myself, what other cancers are there like cervical cancer for which early detection saves lives, for which that's not controversial? The most deadly cancer is lung cancer, and at that time there was evidence that early detection cost lives because you'd be cracking ribs, opening people up, and that surgery itself had a high mortality rate. The second most deadly cancer is colon cancer. Colon is huge. It's 5 feet long. It has a lumen when you open it up that's 8 inches across, so an 8-inch circumference. It has the highest rate of cell replication in the body. And when you eat things, everything that you eat may be a potential carcinogen, cause a mutation. So it's an organ, because of what it is in contact with, that is ripe for mutations and cancers developing. Colonoscopy had been demonstrated to reduce mortality by finding polyps. Just like the pap smear detects pre-malignant cervical lesions, colonoscopy can detect pre-malignant colonic lesions. The problem with a colonoscope, as any of you who had one understand, I did one for my market research. I was in my 40s. And this is not, it's important to be able to speak with conviction, but the worst part's the prep. I was doubled over on the floor with cramps because I somehow thought that the gazpacho I had earlier that day was a clear liquid. It is not. And the bowel prep doesn't go well with gazpacho. But also, when you insert the scope, it distorts and distends the colon, which is highly innervated. And so the procedure needs to be done under sedation. And that means that it takes 2 people who lose pretty much a day to get the procedure done. Compliance then and now with colonoscopy is low because it's not really a procedure meant to be used for screening medicine. A pap smear is. It takes less than a minute. This is not. This is an adventure. So this was the approach that we had, Mr. Stooley. And we thought that the stool is a perfect collector of cells. And so to us, the comparison to the pap smear was clear. You get cells from the cervix and examine them, look for big dark nuclei. You do the same from stool, and this is an engineering problem of cleaning up a stool sample is only incrementally harder than cleaning up a cervical sample. Both have blood and mucus. Here there's additional fibrous matter and bacteria. But it was an engineering problem. We figured out how to transport stools in a way that's not offensive and to process them in a way that's not offensive. And we thought the cells might be preserved and we could do the equivalent of a pap smear of stool. That didn't work. Cells are lysed by something in stool, but the DNA is released. The DNA, if it has mutations, can be detected. It's a tiny part. The DNA that's mutated is a tiny part of the entire sample, so it's a weak signal detection problem, an engineering problem. We overcame the specimen handling problems through engineering, and we set out to look for mutations in DNA. The reason we did this was because the work of one of our scientific co-founders, a guy named Bert Vogelstein at Hopkins, a guy who is a historic figure in understanding cancer. What we know today about cancer proceeds when mutations accumulate in a single cell over many years is Bert's work. He and his colleagues at Hopkins have changed the way we think about cancer and changed our approaches to early detection and to treatment. What Bert showed was that for early lesions, pre-malignant lesions, so-called adenomas, they were pretty much a defined set of changes that if you could detect them in the stool in minute amounts, you could do early detection. As the malignancy progressed, most notably mutations in a gene called p53, it became harder to cure, and usually p53 mutation signaled lymph node involvement and poorer outcomes. We liked the idea based on Bert's work, of looking at stool because stuff from the adenocarcinomas shed into the fecal stream, we thought years before they would shed into blood, and so this would be a good target. We wound up with a lot of IP on methods of preparing samples, methods of isolating abnormal DNA and detecting mutations, and it took 3 tries. You know, if you're a software person, you rewrite a line of code, that's an experiment. If you're a hardware engineer, electrical engineer as I am, you turn the knob and it's an oscilloscope, that's an experiment. In medicine, a clinical trial is an experiment. From planning to conclusion is 3 years, and then figuring out what went wrong is usually another 2 or 3 years. It took 3 tries to wind up with these results, which were published in the New England Journal in 2014. The current version of Cologuard, as it's called, finds cancer at a rate, even early cancers, at a rate comparable to colonoscopy, and finds advanced adenomas, premalignant lesions, at a rate of about 70%, which is enough to have epidemiological impact and far better than any other non-invasive method. Indeed, there too, comparable to colonoscopy, because polyp finding for colonoscopes isn't that easy. So things here turned out well. The company got FDA approval around the time that article appeared in the New England Journal. And under a guy named Kevin Conroy's leadership, Exact has become the fastest growing company in the history of the diagnostics industry. A few reasons for that. I would say, of course, big market, clear value proposition, strong IP. Kevin would say it's the television advertising, that too, which itself was an innovation. What's interesting, if you have a drug and you advertise on television, you know, all those scary side effects and rare deaths have to be disclosed, but not if you advertise a diagnostic. You can do ads that are actually humorous. The company today, right? The company today has a market cap of about $13 billion, and the story isn't over yet. It is becoming a standard of care. It is certainly displacing colonoscopy for screening. It has led, I'm proud to say, a decline in the number of fellows signing up for gastroenterology as a subspecialty. Because right? This is a pernicious application. Screening is a pernicious application of endoscopy, and I'm glad to see that it's on the decline. On the other hand, when patients are positive for the Exact test, the predictive value is in fact very high, that a subsequent colonoscopy will find something and the physician can treat the patient, and a life may well have been saved. So those were two successes. This talk wouldn't be complete without a discussion of a failure. I talked at greater length about this last year. It felt afterwards that I had combed myself, my colleague Teresa Tribble, who may be here, and I kind of combed ourselves with hot combs, but it was the first time I actually publicly talked about a failure. Usually one gets invited at these events to talk about successes. It was cathartic to talk about a failure. The same drive to have impact on early detection of cervical cancer and colorectal cancer was the drive behind doing early detection of autism. Autism is a disorder that affects 1%, 2% of all children for reasons that no one knows. The rate of diagnosis has been increasing. It's a tough diagnosis to have in a family. Symptoms, in retrospect, present as early as 18 months. But the average age of diagnosis in the US remains, despite much increased awareness, somewhere in the +4, age 4 and older, sometimes detected only in first grade. And the earlier that detection takes place, the better the outcome. So we had known some folks at Children's Hospital here and other investigators at other institutions who had one way or other looked at biomarkers, looked at markers not of mutated genes, because only a small fraction of autism has been associated with hereditary passing on of genes, but altered levels of RNA, by implication metabolites, perhaps by implication other proteins. We licensed the technology from Children's. We did collaborations with other investigators that had also worked in the field. And we had predefined, Teresa had predefined levels of success. This is called a receiver-operator characteristic. Think of the green as it works really well, the yellow is kind of okay, and the red is give the money back. We did an initial study and sample collection. We did what was then the largest study, prospective study in autism in its day, which, believe it or not, was only 800 samples. And from those cases, we saw that even just on the training data, the first 500, we could not replicate the work that the academics had published. There were differences between them, so this isn't saying, see, those guys committed fraud, but we couldn't replicate the work. RNA was a poor marker. We tried DNA. We did less well than published results. And then finally, we tried metabolites, where there were also some interesting published results. And we were unable to replicate those either. And we think the reason is because our study was actually quite rigorous. Our patient selection represented the community. And in retrospect, those studies all had methodological flaws in them, primarily in patient selection. So we did say, if we don't hit our 60% or so threshold, we're going to give the money back. And that's what we did. And I have to say that when I started each of these companies, I had no prior sense of one's going to be a blowout success and the other a failure. I felt each was highly risky. And only in retrospect can I tell the story that I'm telling you today. You would have heard me in the 1980s and 90s and early 2000s. I would have been always tentative about where we stand. I wanted to talk a little bit about what I learned in this odyssey. These days, I'm actually not a working guy. I'm on boards of directors. I'm not an active CEO. And so it's an opportunity for me to take a look back. When things succeeded, it's because of that big market, clear value proposition, strong IP. To this day, Cytek has no meaningful competitors. To this day, Exact has no meaningful competitors for non-invasive detection, each for non-invasive detection of important malignancies. If you have a great problem, you can attract great people. You can attract great investors. And the other thing, this is a terribly arrogant thing to say, but I'll say it because I suspect it's true for many of you here. It's a small n, 2 successes, 2 failures, so it's easy to overfit data. But I would say the things for me that worked out best when I and my colleagues were the inventors. We knew what the soft spots were. We licensed stuff from universities and those companies because we needed to well, have freedom to operate. But the fundamental inventions were ours at Cytic and at Exact. When they failed, the companies still were consistent with the credo. They were big markets, clear value proposition. We had strong IP. But a nice way to say it is the science was early. A not so nice way is to say it is we didn't understand the science at the time we committed to the business. It looked like it might work, but in retrospect, we didn't know the data intimately, though we thought we did. It wasn't for a lack of trying. Maybe it was for not being smart enough. And finally, there's a particularly bad thing to do, which is go public too early. I've been a founder myself of 2 public companies and a board member of a company that was private early stage and then went public. And when things are not happy, it's because these are all pre-revenue companies, promising, exciting technology, pre-revenue. That transition from promise and pre-revenue tends to crash when revenue starts unless the revenue runs across some insane ramp. And in medicine, that happens, but it doesn't happen all that frequently. So in retrospect, what I advise companies now is wait until you have a predictable revenue stream and have your company valued on revenue if you're contemplating an IPO. I wanted to talk about 2 other things. One is fundraising and the other is getting out of the way. So what I really enjoyed was the stories that Rodney, that Joy, that Keenan, and that Jeff told. These were great stories. This is the thing we all aspire to. This is a level of storytelling that transcends business. These are compelling stories. I have this really 2 ways here. Tell a good story and don't be boring, because I get to listen to a lot of pitches, and they're mostly boring. And boring is I've got a slide for the market, I've got a slide for the competition, I've got a slide for the business growth, but it's not a story. What we heard here today were great stories. They were funny, they were serious, but they all had loft. They were personal, they were not personal, but they were all great stories. This is the essence of fundraising. Yet, remember that VC is not a science project, and that people who invest their own money, or even more stringently, other people's money, want to see how they'll ultimately make money. And I think if there's any one thing missing in the many presentations I see, I live in Colorado now, and so I get to see a lot of presentations from the university. Fabulous science, difficult to understand the connection between the science and business. And even if it's one slide for a seed stage capital deck, the discipline of connecting scientific innovation with impact. And again, impact can be measured, should be measured for investors in return, but it also can be measured in medicine, in lives saved, in misery avoided, in costs saved. In one sense, it is important to be the smartest man or woman in the room. You have to do your best at knowing everything. But if you don't, say, well, I don't. So you have to both be immodest and very modest at the same time. There's 2 thoughts that are hard to keep in one's head at once. If you can't raise money, it's not because you're not handsome enough or beautiful enough or anything like that. It's because the story isn't a good story, or the value proposition isn't a good value proposition. For each of the things I've done that has worked out or not worked out, I looked at many other opportunities that I let die because, well, they didn't sell. I could convince myself it's a good idea, but if I can't convince others, it's not a good idea. I am a founder. I'm an inventor founder guy. I think I'm at, above average skills doing those things, and I have highly ordinary skills as a CEO. So for me, a question has always been, when do I get out of my own way? Whom do I hire, and how do I assure myself that the person I hire will do a better job at me going forth? And here too, my track record is mixed. But learning what your team thinks of you, that's a good way, that's an important lesson. Learning how to do that using modern HR tools is important because it lets you know when the time has come for a transition. Setting your own milestones, for me, I've learned, is important. If this, and this happens in the company, I'll start looking for my successor. And by initiating that, I don't have the board force it on me. I can lead the process, and have a sense of, which is important to me, of being in control for the transition. And that means I've predefined an exit path, and I can leave on my own terms. So how do I feel about all this? Well, my children have grown up. You know, 2 have turned out really well. The companies aren't mine. They're run by different executives now. But I do feel a deep sense of ownership. And this journey for me, as I think it will be for all of you, win or lose, mostly win, I suspect, has been enormously gratifying. The 4 tough tech companies that I've done are very different than the first 2 that I did. They were not tough. Again, they weren't gratifying. And there are many ways that gratification comes. You know, the obvious ways are economic. The obvious ways are, other obvious ways are, well, seeing your technology be adopted. Here's one I never contemplated. This is the prototype of the ThinPrep at the Smithsonian Museum. It's the National Museum of American History, if I have that right. It's really quite an astonishing thing. I was really quite surprised. So 30 years in tough tech, enormously gratifying. Thank you.